Hey guys, Mr. P. In this video we're going to cover the light-dependent reactions in as much detail as possible. In our last several videos we've talked about how the photosynthetic equation looks, we've talked about the reactants and products, we've talked about the difference between absorption and action spectra, we've talked about the specific pigments that allow plants to absorb the light that they do, and then we covered some different experimental techniques on how to effectively measure the rate of photosynthesis. And so with all of that previous knowledge, we're going to dive in and kind of apply the information we've learned in previous videos to this particular situation, which again is the light-dependent reactions. This particular video is going to cover quite a few objectives, C139, 10, 11, 12, 13, and 14. C139 talks about the photosystem specifically as arrays of pigment molecules that can generate and emit excited electrons. C1310 talks about the advantages of the structured array of different types of pigment molecules in each of the photosystems. C1311 talks about the generation of oxygen as a waste gas from photolysis in photosystem II specifically. C1312 talks about ATP production by chemiosmosis in the thylakoids. This form of ATP production is slightly different than the form of ATP production in cell respiration. This one specifically is photophosphorylation. We'll cover that in this particular video. C1313 talks about the reduction of NADP by photosystem I, specifically producing NADPH. And C1314 talks about thylakoids as systems for performing the light-dependent reactions of photosynthesis. The thylakoid is the location of this particular reaction. So the first thing we need to talk about when identifying all of the necessary components of the light-dependent reaction is the location of the light-dependent reactions. We know that photosynthesis exists within and around the chloroplast within eukaryotic plants. However, it is important to note that cyanobacteria are prokaryotes that exist as a single-celled organism, but they have adapted to utilize the photosynthetic pathway. It is slightly different than what eukaryotes do, and we're not going to focus on their version of photosynthesis in this video, but just a good distinction to make that prokaryotes can photosynthesize in the case of cyanobacteria, but for the sake of this video, we're going to focus on eukaryotic photosynthesis within plant cells. Within all photosynthetic plant cells, there are chloroplasts, that is the organelle responsible for photosynthesis. Within the chloroplast, there are some key structural things to kind of key in on. Chloroplasts have two membranes. They have an outer membrane and an inner membrane. Inside of that inner membrane is a fluid known as the stroma. The stroma is the site of not only the Calvin cycle, but a lot of other enzymatic reactions. Then there are these flattened discs called thylakoids, and those thylakoids are oriented in stacks of those discs called grana. Like we've talked about in previous videos, these discs need to be flattened in order to decrease volume and increase surface area. They serve as little solar panels for the plant, and the whole reason plants have these is to absorb light, and that light is coming from the sun or artificial lights, but it is in the form of photons. If we zoom into the actual cross-section of one of those flattened discs, you'll see that there's a lot of actual biology and biological components embedded within the membrane of that particular thylakoid. You have what is called Photosystem II, an electron transport chain, Photosystem I. You have some other enzymes, light-absorbing pigments, ions, water, chemiosmosis, and ATP synthase. Now, we're going to get into all of this in more detail later, but it is important to note that when we talk light-dependent reaction, we are talking about structural pieces that are embedded within this thylakoid membrane, and this thylakoid membrane is embedded within this chloroplast, which is the organelle. So, the light-dependent reactions capture light energy in the form of photons and will use that light energy to produce NADPH and ATP. Notice NADPH and ATP are coming out of the light-dependent reactions which occur within that thylakoid membrane, and they are going or transitioning from light-dependent reactions to the dark reactions, which is the Calvin cycle, which occurs in the stroma. The light-independent reactions take the ATP and NADPH and they use those in the stroma to produce glucose, which is the purpose of photosynthesis. In the process of doing this, you'll notice that there are some reactants, H2O and CO2. Those are reactants. Those are coming in to not only our light-dependent reactions, but also our Calvin cycle. And then we produce some products. Obviously, glucose is the product we want, but O2 is a byproduct. Now, O2 is going to be released from all plants on the planet as photosynthesis takes place. This O2 has led to an increase in the O2 concentration of the planet's atmosphere, and is the reason the atmosphere now contains a large abundance of oxygen for not only you, but all other heterotrophs on the planet. So what are the advantages of the structured array? When we talk about structured array, we're talking about the pigments that exist within the actual photosystems themselves. In a last video, we talked in depth about the photosystem specifically, and I said that there are a lot of individual pigments that absorb these light photons and then will shuttle them or bounce them around like a laser on a series of mirrors and ultimately shuttle them to a specific special point in that photosystem. That specific special point is called a reaction center. There are chlorophyll A molecules that exist within the reaction center, and the whole point of those reaction centers in both photosystem 2 and photosystem 1 is to take all of the light energy that is being absorbed by the photosystem as a whole and focusing it on a particular point for the purpose of exciting electrons. They will take electrons from an electron donor and they will excite them and then give them to an electron acceptor. There are all kinds of accessory pigments. You'll see that there is chlorophyll A in the reaction center. There are accessory pigments like chlorophyll B, carotene or carotenoids, xanthophylls, anthocyanins, phycocyanins, etc. All of these pigments, you can see, and we've seen this type of graph before, is that all of the pigments are really good at specializing at one particular frequency of visible light. Remember, the plant utilizes visible light, and so that runs from red all the way to violet, and all of these pigments in the photosystems are going to be used by the plant to absorb as much of the visible light spectrum as the plant can. Because the plant utilizes a variety of pigments and they use this structured array and this antenna complex, it allows the plants to be a lot more efficient while doing photosynthesis. So, what does this photosystem look like? Photosystem 2 comes first, and photosystem 1 comes second. You'll notice that they look almost identical. There are some key differences. Photosystem 2 comes first, photosystem 1 comes second. They are both embedded in the thylakoid membrane, which is that small disc inside the chloroplast. And the first thing that's going to happen is a photon from the sun or from artificial light is going to be absorbed by the array and by the pigments and accessory pigments, and it's going to be shuttled. Notice that in this diagram we see the red spectrum and the blue spectrum. Photons emitted from the sun are going to be collected and absorbed by the pigment. They're going to be shuttled around all of these different pigment molecules and are going to be focused on the reaction center of photosystem 2. Photosystem 2 contains chlorophyll called P680. So that's chlorophyll A, P680, and so we refer to reaction center of photosystem 2 as P680. The excited electrons, which are going to be sitting here at this P680 spot that come from photolysis, is going to be energized and is going to be captured at a high energy state by the primary electron acceptor. And water is going to be the source of those electrons. So one of the things the plants need is water, and when we talk about the photosynthetic equation, we know that 6 CO2 plus 6 H2O yields, in the presence of light and chlorophyll, C6H12O6 plus 6 O2. Water is one of the reactants of photosynthesis, and so this is where that reactant comes into play. Water is constantly brought into the plant from the soil through the roots. And as that water is brought into the plant, it's going to be split in a process called photolysis. What that means is it's basically hydrolysis utilizing light as an energy source. And so water is split And when you split water, an H2O molecule, the components of that molecule would be 2 electrons, a half O2 or a single oxygen atom, and 2 hydrogen ions. All of these are going to come into play, but for now, electrons are the important bit because that's what's actually being utilized by this particular photosystem. The hydrogen ions are going to be funneled to the chemiosmosis or the photophosphorylation bit, but oxygen is going to be released as a byproduct again to the atmosphere, and one of the reasons why our atmosphere has accumulated oxygen over time. So as those electrons that get pulled out of water get moved to this reaction center, the photons that are being absorbed and bounced around and focused on this reaction center are going to take those electrons to a high energy state and then give them to a primary electron acceptor. Once they move from the reaction center to the primary acceptor, excited electrons pass from the primary acceptor down the electron transport chain, which is this. We'll call that the ETC, electron transport chain, losing energy at each exchange. These are just a series of redox reactions. When this primary acceptor gains electrons, it's said to be reduced, and then it's oxidized, and this protein is reduced, and then it's oxidized, that protein is reduced, and so on and so on. And when those electrons are on their way to the cytochrome complex, they go through this particular protein, which is called plastoquinone, and then goes to the cytochrome complex before it enters this area of the thylakoid membrane, which is known as photosystem I. Once the electrons get down this entire electron transport chain, they are very low energy because they have given off energy gradually at every step, and so they have to be recharged. At this point, photosystem I, the same photons that are being emitted from the sun are going to be collected downstream, so to speak, by the pigments of this particular photosystem. The same thing's going to happen. All of those photons are going to be bounced around from pigment to pigment to pigment to this particular reaction center, which serves the same purpose. The chlorophyll A in this particular reaction center is called P700. And when those electrons end the electron transport chain, they get placed on that reaction center to be recharged. And when all of the light energy or the photons are being sent to this reaction center and meet the electrons get brought back to a high energy state before being accepted again by a primary electron acceptor. And then once accepted by that primary acceptor, they are going to then be passed down a second electron transport chain, which involves the carrier ferredoxin, before entering an enzyme called NADP plus reductase, which has one job, and that one job is to reduce NADP plus. NADP plus is going to take a hydrogen ion that is floating around in this space that was split off of water, as well as the pair of electrons that just went down this electron transport chain to become NADPH. So NADP plus this hydrogen ion plus this pair of electrons is going to convert, utilizing this enzyme, into NADPH. NADPH is going to be off to the Calvin cycle. That is one of the reactants of the Calvin cycle and is one of the products of this particular system. That brings us into a discussion of photophosphorylation, which is putting all of it together. I know we've just talked through photosystem II all the way through photosystem I, but now you'll notice that we have another player in this space, ATP synthase. That is the enzyme that actually produces the ATP. Remember, in our first slide, we talked about the two things that the light-dependent reactions are making, NADPH and ATP. And so this is the entirety of the light-dependent reactions, at least from a photophosphorylation standpoint. And so to put it all together, the first thing that happens is light emitted from the sun is going to strike the pigments of this particular photosystem, PS2. That light is going to be shuttled around and funneled to the reaction center, which is P680, energizing the electrons that were just placed there during the photolysis of water, meaning water is split into three components. Water is going to be split into electrons, oxygen gas, and hydrogen ions. The electrons are brought to a high energy state before they enter the electron transport chain. Once they enter the electron transport chain, they're going to do a series of redox reactions, releasing energy gradually at each step. that energy that is released by the electrons is going to be utilized to actively transport hydrogen ions from a low H plus ion concentration side to a high H plus ion concentration side. This is our low side, this is our high side. Why is this side the high side? Because this is where photolysis is happening. If photolysis is happening on this side of the membrane, and one of the products of photolysis is the hydrogen ions being released, you would expect there to be an accumulation of H plus ions on this side of the membrane. And in fact there is. This is what we call the thylakoid lumen, which is the space inside of the disc. This would be the chloroplast stroma, which is the outside of the disc. So as water is split and H plus ions accumulate in this lumen space, some of those H plus ions will flow through ATP synthase, which we'll come back to. But as they flow through, they move to this low side, and in order to keep that concentration gradient existing, we have to take the H plus ions from this low side and pump them back to the high side. That is an active transport process, and that requires energy. The energy comes from the electrons, which we being plants, from the sun to begin with. So as the sun is absorbed by the pigments in photosystem II, it energizes these electrons. The electrons move down the electron transport chain. It actively transports the hydrogen ions back to the high side. When it gets to the end of the electron transport chain, the electrons are low energy and have to be re-energized, and so they are moved to reaction center P700, which is the reaction center of photosystem I. Light is absorbed by the pigments of photosystem I, which are shuttled around and focused on that reaction center. That reaction center, P700, will re-energize these electrons before sending them to a secondary smaller electron transport chain. Once those electrons end their second smaller electron transport chain, they end up at that NADP reductase enzyme, which has one job. Remember, it takes NADP, H plus ions, and electrons, and it adds them all together to produce one reduced NADP molecule, which is NADPH, and that NADPH is off to the Calvin cycle, which, remember, happens in the stroma. So that is one of the reactants of the Calvin cycle. We just made it utilizing H plus ions, electrons, and NADP plus. Once those electrons are gone, we're going to have a constant flow of those electrons, or new electrons, as more and more and more water is split. Now, we focus our attention on the H plus ions. As the H plus ions begin accumulating on this side, again, this is our high H plus ion concentration, those H plus ions are going to flow down their concentration gradient passively. So this is a facilitated transport mechanism. It doesn't take energy, and so those H plus ions are just flowing down their natural concentration gradient. So they go from an area of high to an area of low. Those H plus ions are going to flow through ATP synthase, and as 3 H plus ions move through ATP synthase, that's going to be enough energy for this enzyme, ATP synthase, to synthesize or produce one molecule of ATP. ATP, again, is adenosine triphosphate, and this enzyme takes an ADP, adenosine diphosphate, takes an inorganic phosphate and smashes it onto that ADP, turning ADP into ATP. That is called phosphorylation. That's how we get our photophosphorylation process. Photo means light, phosphorylation means to phosphorylate. This particular process is phosphorylating ADP utilizing light as an energy source. ATP is made. It too is shuttled to the Calvin cycle, which takes place in the stroma. These are the two reactants of the Calvin cycle, and they are the two products of the light-dependent reaction. So light-dependent reaction occurs in the presence of light and will utilize H2O to produce NADPH and ATP. In the process of producing these two reactants for the Calvin cycle, it produces a byproduct in oxygen gas that is released and helps contribute to our atmosphere. That's it for this video. If you learned something, give it a thumbs up. If you have any questions at all, leave them in the comments. Subscribe to the channel. We'll see ya.